the fifth step is the non-electrogenic function. The first one is the release of the neurotransmitter. The second one is the action of the neurotransmitter in the post-junctional receptor. Third one is the dissipation or dissociation of the neurotransmitter. And the fourth one is the initiation of postsynaptic action. And fifth one is the non-electrogenic function. Let us see what is this non-electrogenic functions. The continual quanta release of the neurotransmitters in amounts may not be sufficient to elicit a post-junctional synapse. Probably is most important in transjunctional control of the neurotransmitter action. See here, many times the neurotransmitter is released from one synaptic cleft, may get diffused into the other, and the activity and turnover of the enzymes involved in the synthesis and inactivation of neurotransmitters, the density of the postsynaptic and postsynaptic receptor, pre and postsynaptic receptors, and other characteristics of the synapses probably are controlled by trophic actions of the neurotransmitter or other substances released by the neuron or the target tissue. So usually what happens, what is the non-electrogenic function means, here the electric current, especially sodium and potassium current, they are not involved. Some of the enzymes may be involved in the post-junctional activity, and also the presynaptic and postsynaptic receptor density, total quantity of the density, these are all important. And sometimes in 90% it's okay, that's the electrogenic function is there. In few, non-electrogenic function may also be there. And the next terminology is the co-transmission. Co-transmission is most peripheral and central neurons on stimulation have been shown to release more than one active substance. In the previous slide, you have seen that, especially in the central nervous system and adrenergic and cholinergic nervous system, along with the acetylcholine, norepinephrine, and GABA or glutamate, some other materials are also secreted, which helps the primary neurotransmitter to have the action. So along with this thing, especially this acetylcholine and norepinephrine, purines like ATP and adenosine, they are also released, and the peptides like VIP or neuropeptide Y, NPY, and substance P, enkephalins and somatostatins, they are also released in certain places. And also the nitric oxide and prostaglandins, they are released along with the acetylcholine or norepinephrine, depending upon the type of the nervous system, sympathetic or parasympathetic. And all these are known as co-transmitter. Co-transmitters are the one which are released with the primary neurotransmitter and have the action with that of the acetylcholine. And the examples are the purines, ATP, adenosine, and the peptide VIP or vasoactive intestinal polypeptide, neuropeptide Y, especially in the CNSC, and substance P, enkephalins, somatostatin, all these things, and nitric oxide. So, and prostaglandins, these are all called as the co-transmitters. Co-transmitters are the one which are secreted along with the main neurotransmitter, that's acetylcholine or norepinephrine. And in most of the autonomic cholinergic neurons, and the ATP is secreted with acetylcholine and norepinephrine. So the VIP is also associated with acetylcholine, whereas the ATP is associated with both acetylcholine and norepinephrine. So the transmitter at some parasympathetic site is nitric oxide, and these are called as the nitrergic nerves. So wherever the co-transmitter is nitric oxide, the nerves are called as the nitrergic nerves. The co-transmitter vasoactive intestinal polypeptide or VIP of parasympathetic neurons produces a slow and long-lasting response, while nitric oxide has an intermediate time course of the action between VIP and ACH. So, the role of the co-transmitter is to either slow or long-lasting response, to create the slow or long-lasting response wherever it is required. Similarly, in sympathetic neurons, the co-transmitter neuropeptide Y is slower acting and ATP is faster acting than the neurotransmitter, and many times the co-transmitter itself will be more potential or more inhibitory in nature depending upon the type of the nerves. Co-transmitters like nitric oxide, VIP or the NPY diffuse to a wider area and can affect the receptors at some distance from the site of release. So, the action of the especially the neurotransmitter is little bit complicated because of the co-transmitters like nitric oxide, VIP and NPY, because sometimes their action is more than that of the original neurotransmitter. The co-transmitter's action will be more when compared to the original transmitter. And this picture also reflects what is a co-transmitter and the primary transmitter. The red one is, this is the primary transmitter and this is the co-transmitter. So, in the synaptic vesicles, the both are stored. R1 transmitter receptor here and R2 this co-transmitter receptor. So, on the post-junctional membrane, the receptors are there for both the primary transmitter as well as the co-transmitter here. You can see maybe a adrenergic system or cholinergic system. This is common. There will be separate receptors for the co-transmitters and also for the regular neurotransmitter or primary transmitter. So now we have what you call as learnt about the general neurotransmission, neurohumoral transmission. Now we have learned. That's the first one is the axonal conduction, followed by the junctional transmission. Junctional transmission is classified into 5 steps. The number 1 is the release of the receptors into the presynaptic terminal, then the initiation of the action of the post-synaptic end. The second one is the combining or junction of the neurotransmitter with that of the postsynaptic receptor. Third one is destruction or dissipation of the receptor, the neurotransmitter, and the fourth one is initiation of the post-junctional activity. Fifth one is the non-electrogenic function. So these are all the sequences how the neurotransmitter occur, and we have to learn it with the system wise how this occurs in especially in the cholinergic transmission and also the adrenergic transmission. So coming to the neurohumoral transmission of the cholinergic nerves, how it is going to occur. So all the steps will be followed here, starting from the axonal conduction, from the junctional transmission. So acetylcholine is a major neurohumoral transmitter at autonomic, somatic as well as the central side. So acetylcholine is a ubiquitous or universal neurotransmitter which is also present on the skeletal muscles and also the what you call as the central nervous system and in the parasympathetic, sympathetic and parasympathetic, both division it's present as preganglionic neurotransmitter. So there are varieties of the enzymes are there. Then the first step is synthesis of the neurotransmitter. That's the synthesis of acetylcholine here. How it is synthesized? There are 2 very important enzymes like the choline acetyl transferase and acetylcholine esterase are involved in the synthesis and degradation of the acetylcholine. So The cholinesterase transferase is involved in synthesis of the neurotransmitter, whereas the acetylcholinesterase is involved in the synthesis degradation of the acetylcholine. So first let us discuss about the choline acetyl transferase. What is its action? So choline acetyl transferase catalyzes the final step in the synthesis of acetylcholine, the acetylation of the choline with acetyl coenzyme A. So here the choline and acetyl coenzyme A, they are going to form the acetylcholine, and this step is being catalyzed by choline acetyl transferase enzyme. And acetyl-CoA is derived from pyruvate via multi-step pyruvate dehydrogenase action, or it's synthesized by acetate thiokinase. This acetyl coenzyme A is very much required for the synthesis of acetylcholine, and it is derived from the pyruvate due to the multi-step pyruvate dehydrogenase enzyme and the acetate thiokinase enzyme. And this catalyzes the reaction of acetate with adenosine triphosphate to form the enzyme bound acyldenate, that's called as the acetyl AMP. Ultimately, this is going to cause acetyl AMP. Then, what happens in presence of CoA? In presence of the CoA, trans-acetylation and synthesis of acetyl-CoA occurs, and it is produced. And with the help of the choline acetyl transferase, like other protein constituent of the neuron, so is synthesized. This cholinesterase transferase is synthesized within the perikaryon and then transported along the length of the axon to the axon terminal. So to simplify this thing, the choline acetyl transferase and acetylcholinesterase both are existing in the vicinity of the nerve, whereas the choline acetyl transferase is present in the exoplasm, the acetylcholinesterase is present in the synaptic cleft. The synaptic vesicles appear to be formed at terminal and axonal terminals contain a large number of mitochondria where acetyl-CoA is synthesized. So, to form acetic acid and choline, to form the choline and acetyl-CoA, then the choline need to be uptaken and acetylcholine need to be synthesized. So choline is taken up from the extracellular fluid to the exoplasm. So please imagine that the neuronal terminal, the choline is taken up from the extracellular fluid into the exoplasm by active transport, because choline is a little big molecule. Because of the active transport, it enters the exoplasm, then it combines with that of the synthesized CoA with the choline acetyl transferase enzyme. The final step in the synthesis occurs within the cytoplasm, following which most of the acetylcholine is sequestered within the synaptic vesicles. Then after this synthesis of the acetylcholine, they are stored in the vesicles, and potent inhibitors of the choline acetyl transferase have no therapeutic utility in part because the uptake of choline is the rate limiting step in the biosynthesis of the acetylcholine. What's a rate limiting step? Is the one which affects the further neurotransmission. It's a step in which varieties of the chemicals are released or synthesized or uptaken. And this, if we cut this particular rate, then there should be effect on the neurotransmission. But this is not so happening in case of the choline acetyl transferases. So that's why they are telling that the acetylcholine cholinesterase transferase agonists or antagonists has no therapeutic utility, because the uptake of choline is the rate limiting step in the biosynthesis of acetylcholine. And after synthesis, see, after this particular acetic acid, this acetyl-CoA and the choline, they are combined to form acetylcholine, then they are transported in and stored in the synaptic vesicles. And here one of the drug we have to remember, it is a vesamicol. Vesamicol inhibits the transporter system. The drug blocks evoked release of acetylcholine without affecting the influx of the calcium into the nerve ending that the choline transporate or acetylcholine synthesis. That's why the acetylcholine synthesis will be inhibited by the vesamicol because it blocks the release of acetylcholine, but it is not going to affect the calcium ions in the nerve ending. And of course, the choline transporate or acetylcholine, all these three are not affected, just it is going to inhibit the release of acetylcholine. And the second enzyme, the first enzyme is the cholinesterase transferase, and second one is the acetylcholinesterase. This is one of the most important enzyme therapeutically to us. See, in case of organophosphorus toxicity, the acetylcholinesterase enzyme is irreversibly inhibited. That's why lot of acetylcholine is going to accumulate in the synaptic cleft and cause lot of convulsions or the postsynaptic activity. And at the neuromuscular junction, the mediator must be removed from synapse almost immediately with just like flash-like suddenness. Just a flash means you have seen the camera flashes within a fraction of seconds. This need to be terminated. The action of the acetylcholine is need to be terminated. That's called as the flash-like suddenness, especially in the cholinergic system. The removal of the neurotransmitter is just flash-like with the help of the acetylcholinesterase. So, modern biophysics methods have revealed that the time required for hydrolysis of acetylcholine is lesser than a millisecond. See, a second is divided into 1000 milliseconds. So, less than a millisecond is required to remove all the released acetylcholine in a flash-like suddenness, that you should remember. Acetylcholinesterase is found in cholinergic neurons, maybe dendrites, pericardia and axons, and also in the vicinity of the cholinergic synapse and in other tissues wherever the cholinergic innervation is present. Means if you want to ask where this acetylcholine esterase is located, means the cholinergic neurons may be in the dendrite, a branch of the nerve body, then pericardia and axons. So it is highly concentrated in the neuromuscular junction. Of course, the acetylcholinesterase is very high in concentration in the neuromuscular junction. That's why the acetylcholine will be destroyed immediately after its release, especially in the neuromuscular junction. Or else what happens is there will be fasciculation, continuously twitching up of the muscles, then the flaccid paralysis may occur. Especially this happens in organophosphorus compound toxicity, and hydrolysis of the acetylcholine occurs in immediate effect, especially the plasma and liver. So this hydrolysis of acetylcholine occurs in immediate vicinity of the nerve ending. And another enzyme is there, that's the butyryl cholinesterase. There are three enzymes. The number one is the cholinesterase transferase, which makes the acetylcholine synthesis, whereas the acetylcholinesterase is the one which is having the destruction action of the acetylcholine into acetic acid and choline, whereas the choline is taken up. The another enzyme, butyryl cholinesterase, it's also known as the BUCHE, or it's also known as cholinesterase CHE or pseudocholinesterase. The butyryl cholinesterase is also known as pseudocholinesterase. It's present in various types of the glial satellite cells, but the action is very, very limited. So what is the action of this butyryl cholinesterase? So it is present primarily in the plasma and liver and cause hydrolysis of ingested esters from the plant sources. So the reason being the presence of the butyryl cholinesterase is every day we are consuming a lot of what you call as the liver, in the liver and other thing, the hydrolysis of ingested esters, especially the esters of the acetylcholine or other congeners may be there. So the ratios of acetylcholine esterase and butyrylcholine esterase change substantially during development of the nervous system, especially in the nervous system this is present. Acetylcholine and esterase and butyrylcholine esterase are typically distinguished by the relative rates of acetylcholine and butyrylcholine hydrolysis and by effects of selective inhibitors. So acetylcholine esterase inhibitors, of course the butyrylcholine esterase. So whenever any materials, especially plant sources, esters from plant sources are coming, then the butyrylcholine esterase is going to destroy them.